Good morning, their students, their colleagues, they all. Welcome to this fourth and last innovation talk of LUMSA University of this semester. I'm Leonardo De Cosmo. I'm professor of digital transformation at LUMSA University of Rome. Innovation Talks is a cycle of webinars aimed to discuss innovation, technology, sustainability, and the future of business with expert and opinion leader in the business community. Innovation Talks are organized by the Department of Law, Economic, Politics, and Modern Languages, and the Master Degree in Management and Finance in partnership with Association of Alumni LUMSA. And today we will talk with Marco Marcia. Marco Marcia is a chemistry, no? But he works in the biological fields. He works in EMBL in Grenoble. He's a group leader there, and he will explain us as how robotics and artificial intelligence is changing the way we study biology. And in particular, we will talk about Google biomapping of genome. Is it right, Marco?
Yes, exactly. Can you all hear me?
Yes,
well,
of course.
Yeah, so it's a pleasure to be here, and hello everybody from a very rainy and snowy Grenoble in France, close to the Alps. Thanks, Leonardo, for your kind introduction. Leonardo and I met recently and discussed this option of sharing some insights into biology and the technology that contributes to biological studies to all of you. It's an experiment. I count on your contribution proactively to make it happen and as fruitful as possible. It's a challenge, clearly, to bring together biology and management and finances, but try my best to make this topic as accessible as possible to all of you, especially trying to capture your attention and the implications that these studies have for what are your, I expect, future career perspectives. And so I've decided to organize my talk in three parts. I will have to give you at least a basic few slides introduction on structural biology. I'll then move on to explain to you how robotics and artificial intelligence support our studies nowadays. This is really cutting-edge research that is going on. Most of the tools I will present to you were not available even just a few years ago, and I'll try in several slides to make this historic perspective more clear to you by comparing what was available to me at the beginning of my career just 10 years ago and what is now the field looking like. For my presentation, I will use as much as possible and voluntarily a lot of online resources in the forms of videos and movies, and I will refer to online databases and tools that are open access so that all of you can appreciate how much structural biology has become actually accessible also to the audience of non-specialized scientists. And I'd be happy to, you will have this presentation online and recorded, and I'd be happy to share the slides with Leonardo so that even in the future, if you have curiosity or if this talk has captured your attention, you can still go back to those resources and revise, go deeper into the matter, and appreciate it in more details. The only concept of biology that I need to share with you is what is depicted in the current slide. And this corresponds to the most fundamental principle of life sciences. That is the central dogma of biology. This is something that should be reminiscent to you of your studies in high school, where you most certainly have had a flavor of how life occurs in our bodies, in our cells. All of our cells contain genetic information in the form of DNA molecules that have a very specific sequence. This sequence is converted, transcribed is the technical term, into messenger RNA molecules, so another form of these sequences, very similar but transient and suitable to be then converted with a form of translation into a sequence of amino acids that form so-called proteins. Proteins are the fundamental building blocks of our cells. They are the elements in our cell that carry out all the functions that allow us to be alive, starting from providing energy to our body, metabolizing the nutrients that we eat with our food, making it possible for us to see, to hear, to sense the environment and respond to this environment around us. Okay, so we have three classes of fundamental biological macromolecules: DNA, RNA, and protein. And what structural biology does is that it is the branch of biology that allows us to get a visualization, very high resolution visualization of these molecules, to understand how they look like and how they function. And I make the parallelism with Google Maps because it's exactly the same. When you go to a new city, if I come to Rome, I'll have to orient myself. I'll open Google Map to know how Rome, the city, is organized, where I can find the touristic attractions, where I can find the city hall, the pharmacy, the schools, or LUMSA University, whatever I need to orient myself, navigate, and understand how Rome or any other city works. Okay, with structural biology, we do exactly the same, but for biological macromolecules. And to give you a flavor of this, I'll show you a couple of videos of what can be obtained from structural biology studies. And I'm going to show you how, through structural biology work, we have over the years elucidated the mechanism of function of two of the most fundamental processes in life, exactly the ones that I depicted here before: transcription and translation, which are carried out by these two protein or protein RNA complexes, okay? So you can fully appreciate, and you'll find these videos at these references, how biology works and the complexity that structural biology has allowed us to capture for biological systems. All what you see here in these cartoonish representations, these are proteins that assemble together, they move, and they dynamically allow these processes to happen. You see, this is the polymerase, that is the enzyme that reads the DNA sequence and produces RNA sequence. You see the DNA in blue, and soon you will see the emerging chain of the RNA also being slowly produced. And structural biology is what allows us exactly this, to capture all these mechanisms at extremely high resolution, watching atom by atom in action. You see the DNA and the RNA is starting to be produced just now in the core of this enzyme. You see RNA starting to be formed and slowly reading the DNA sequence. Okay. And the same we can see in this other movie, where we can see actually how proteins are produced, this other extremely fundamental biological process. Here, the machinery that catalyzes this process is called the ribosome, and it reads RNA, which you have seen entering into the cavity of this enzyme before. It reads RNA to produce a protein molecule. And all these movies that I'm showing to you derive from years of studies and photographs of the system that we have captured through structural biology in all these intermediate states that we then put together. Okay, so here you see exactly the process happening. The ribosome, the RNA, and elements called tRNAs that bring the amino acids together to make the protein chain being synthesized and being produced. Now, apart from appreciating and understanding at an extreme degree of detail how biology works, structural biology also has an important implication for human medicine, because in the same way as when I come to Rome, I understand how Rome works. If I want, I can, intervene in how Rome is
Once you solve
organized.
the ribosome, it's very easy. Solving
Sorry.
the ribosome
When I come to Rome and I understand how Rome works, I can also intervene on its organization to make it work in a different way. Okay, if I am the mayor of Rome, I can change traffic to make the city work more efficiently. And in the same way, we biologists and biochemists, pharmaceutical chemists, we can tune the function of these macromolecules, for instance, using small molecules. That is the way by which we develop medicines. Okay, and you'll see described here how this process has been done to produce antibiotics that inhibit the production of proteins in specific bacteria. And I let a very eminent scientist, a Nobel Prize winner in the field, explain you this process, Venkira Krishnan.
After having solved it, it's easy to solve how antibiotics bind to it. And so you can do the same experiment and get the image with the antibiotic, and then subtract away the rest of the ribosome. And what you're left with is the image of the antibiotic itself. And so here's an antibiotic called spectrinomycin. It binds in this tiny little pocket here in the ribosome. And because this head is supposed to move during the ribosome function, it kind of locks the movement of the head and doesn't allow the ribosome to to move, and so it stops the ribosome death. And using this method, we solved the structure of a large number of antibiotics.
Okay, so I hope that with this brief introduction, you have appreciated how powerful structural biology is, and also how important it is for understanding human physiology, human diseases, and for controlling these diseases through the development of medicines. I'll explain you now how what is the process that allows us to determine those high-resolution structures of biological macromolecules. And in doing so, I will emphasize how now robotics, online digital resources, and artificial intelligence tools are enabling this process in an easier and more accessible manner. The process is a very long, typically, and tedious process that starts with the production and purification of those proteins that we want to characterize. And then it moves on to preparation of crystals or samples suitable for cryoelectron microscopy. Now, crystallography and cryoelectron microscopy are the two leading structural biology techniques. They are the cameras that allow us to take photographs of biological macromolecules, okay? And these processes imply collecting data, collecting the photographs, and analyzing them to then reconstruct the images of the molecules and the movies that I've showed you before. The first step is the production of high amount of the proteins of interest that we want to characterize from a structural point of view. This process, when optimized, takes weeks, several days to weeks. but typically to optimize the process, several weeks to months are actually necessary. And so while until recently, all these procedures were done manually through trial and error processes, now we have available technology that enables these studies in a more automated manner. And you will listen how this works from these colleagues.
The overall goal of the following procedure is to use a high-throughput expression screening protocol to quantify in E. coli the level of soluble proteins using an automated liquid handling robot. This is accomplished by first inoculating 96 well pre-cultures with cells transformed with plasmids encoding the target fusion proteins. The next day, 24 well plates filled with autoinduction media are inoculated with the overnight pre-cultures to generate expression cultures. After harvest and freezing, soluble proteins from the expression cultures are then purified by immobilized metal affinity chromatography. Ultimately, the solubility levels for each intact target fusion can be measured to determine the optimal expression conditions for protein production and the generation of successful targets.
So although there was some technical language in this movie, I hope you could appreciate the fact that through parallelization and automation, we could shorten down a weeks to months long process into a very compact time frame. And this, of course, makes it accessible and allows, increases the chances of success. Chances of success of producing high quality target proteins, okay, that are suitable for structural biology studies. Suitable for structural biology studies means, for instance, that these proteins can be crystallized to be observed with the camera, as I said before, of X-ray light. Crystallization is a process that is also very long, takes hours to months sometimes, and is a trial and error screening process with the goal of producing these nicely organized little crystals that are extremely small in the micrometer side, in very small drops. Now, this entire process, just 10 years ago, when I started my career in structural biology, had to be done manually. And here you see myself at the microscope handling one of those very tiny drops that you can see here in the center of the video. In these drops, there are crystals, which you see here. And I had to fish these crystals using these special tools that act like a spoon, okay, to capture the crystals of interest. You can imagine that the throughput of such manual approach is very small, is very limited, and most importantly, it is prone to errors because these crystals are extremely fragile. In the last few years, colleagues here on my side at EMBL Grenoble have developed a completely automated technology that allows for such a process to happen robotically. And you will see this represented in this video. You can select those drops at a camera in front of your computer. You see the many crystals. You can tell the robot which crystals you are interested in fishing out. You easily select it through a graphical user interface, and then by pretty much pressing a go button, the robot does the entire job for you. Here you see really the robot in action. You see it going, fishing out these very tiny crystals from the crystallization plate here on this side, and moving these crystals to the X-ray beam under a liquid nitrogen stream for freezing these crystals. Okay, so this is a robot that handles very tiny objects, not bigger than a few micrometers. Okay, what do we do with these crystals? Well, we bring them, we have to image them with X-ray light to produce this sort of very fascinating pictures, which are called diffraction images, from where we can reconstitute the images of our molecules. This sophisticated process happens only thanks to the availability of extremely large infrastructures, international infrastructures that are called synchrotrons. We have one here in Grenoble, just across the street from my office. I'm showing you instead a brief video of what a synchrotron is and what it looks like, taken from the website of the Elettra synchrotron in Trieste, to show you that also in Italy we have such advanced technology.
The Electra laboratory is located not very far from Trieste, on the Carso near the town of Basovizza. It was built to produce an exceptionally bright light, synchrotron radiation, a tool that enables researchers to detect structural details of materials otherwise invisible. This tool has many applications in different fields. Let's have a look at the source of this light with such exceptional features. Everything starts in the booster that shoots electrons and accelerates them at extremely high energy. An electron source is located in the first part of the booster. Here, charged particles are admitted by a metal electrode using a principle similar to that operating in the cathode ray tube of a standard television set. The electrons are guided inside a thin, circular, high-vacuum steel tube of 120 meters circumference through intense electromagnetic fields. They are then accelerated to 2.5 billion electron volt. The electrons are shot like cannonballs from the booster into the transfer line and finally injected into the storage ring, 260 meters long. Here they will circle for 24 hours without interruption, almost at the speed of light, and their energy will remain constant. During this time span, they will cover a distance equivalent to 4 times the diameter of the solar system. Synchrotron light is generated in the storage ring. The ring is, in fact, an alternation of straight and curved sections. In each curved section, a powerful magnet guides the electrons, forcing them to deviate from their path. Each time they deviate, the electrons emit energy in the form of light. The beams of light tangent to the storage ring are conveyed into the beam lines, where the experiments are performed.
So you get the flavor of the complexity of these large infrastructures. And what I want to point out to you is you guys are expert in management, in financing. Here, we want to decrypt the very secrets of life. This comes at a cost of making extremely large investment and is only possible through the cooperation of very large international, supranational communities. Now, we have looked at crystallography as a tool to visualize macromolecules. I mentioned before that there is another technology that is rapidly emerging to enable the exact same process. This is called cryoelectron microscopy. And this really works as a camera to make photographs of our biological molecules. An electron microscope is again a highly expensive instrument, very complex to build. It requires dedicated locations, rooms that are designed specifically to host the highly sophisticated components of this microscope. you have to keep in mind that we want to visualize objects at a resolution below a nanometer. A nanometer is 10 to the 9th meters, okay? And therefore, the technology that we need to use need to be extremely precise, extremely well aligned to give us this detailed insight. With a normal camera, we have a resolution in the range of meters, okay? We have to improve it 10 to the 9th times. That's why the technology behind is so complex. Now, a couple of videos to give you a flavor of what is instead cryoelectron microscopy and how different it is from crystallography.
Samples used for cryo-EM are applied to the surface of the grid, blotted to create a thin layer, and then plunge frozen to immobilize the particles. At the macro level, cryo-EM grids contain a metallic mesh, typically made of copper or gold. The mesh contains an array of holes surrounded by a support material, such as amorphous carbon. These holes support the vitrified particles and are targeted for cryo-EM imaging. At the nano level, the particles are ideally dispersed throughout the hole and adopt a broad distribution of orientations. In reality, many particles adhere to the hydrophobic air-water interface in a limited number of orientations and become partially denatured. A successful cryo-EM project is dependent on the ability to reconstruct 2D projections of a particle into its 3D structure. Negative stain analysis is a relatively simple method that provides a first-order estimation of how well the particles are averaged. Particles should be monodisperse in solution, meaning that the sample solution consists of molecules that are roughly the same mass, shape, and size. These particles are combined into separate class averages based on their structural similarities.
So this is the way we image individual particles, and you can see them one by one. So to recap this intermediate part of my talk, we have two technologies to take high-resolution pictures of biological macromolecules experimentally. And this gives us atomic level insights into how these molecules and therefore our cells, our organism can function and how they can be modulated by drugs in case of diseases. In the last part of my talk, I want to tell you about a recent revolution in the field, because with the advent of artificial intelligence algorithms, all this experimental pipeline that is long, time-consuming, and requires enormous financial resources and human resources to be successful has been partly supplemented or let's say integrated by and facilitated by sophisticated computational approaches that enable us to predict the structure of those molecules, of those biological molecules, and therefore get initial insights of how they may function so that we can better plan the experiments and make use, okay, of the information that we are interested in a more practical and efficient manner. I'm talking specifically about one software artificial intelligence algorithm that has become available through the cooperation between colleagues at Google, at DeepMind, together with colleagues in the academic field from another EMBL site located in England near Cambridge. The European Bioinformatics Institute. So this what I'm going to present to you in the next 2-3 slides before concluding my talk is extremely exciting work that has been carried out by these colleagues over the last just few years, actually few months. For many years, the community has tried to predict structures of biological molecules from their sequence without doing any actual experiment, without taking the photographs of those images. But for many years, the field has not been able to advance towards getting any reliably close images of biological macromolecules. A breakthrough happened a couple of years ago with the advent of this artificial intelligence tool called AlphaFold, and particularly its improvement second generation version, AlphaFold II, which is now able to predict structures of biological macromolecules with extreme and unprecedented accuracy. You can appreciate this by visualizing this rotating movie. What you see here again is one of these cartoonish representation that shows the structure of a protein. And in orange is the experimental structure determined by crystallography. In blue is the structure predicted by the computer. Okay, you can appreciate how closely the two resemble each other. There are a few differences, but typically only located in regions that are already intrinsically flexible. Okay. How did we get there? I'm not a computer scientist, so I'm not going to go in the details of the algorithms, but you can find those of you that are expert in artificial intelligence and programming can find the technical details in this very important seminal paper published last year in Nature, one of the most highly reputed journals for science. It consists of a pipeline that goes iteratively through a series of steps that start with the sequence of the protein X, a target that the researcher is interested to study from a structural functional point of view. The computer takes into account the sequence of this target as well as the 2, 3, 4 decades of past knowledge on experimental structures that have been determined, which are all accessible through open source resources, as well as the sequence of evolutionary related proteins to the one that we want to characterize. With a combination of this information, which we have acquired thanks to many decades of work in biology, now the computer can reconstruct accurately the 3D structure of our target protein. This is a real revolution. We now have, and you can see the resource here at this link, a structure prediction of essentially every single protein that we know exists on Earth. These predictions are extremely high confidence, and the last year and a half of experimental studies have confirmed that the level of accuracy of this prediction is unprecedented and really close to reality. We cover structural information for over 1 million organisms, and all this is accessible to all of you, even from your cell phone. Therefore, this computational resource has now changed the standard pipeline for structural biology, which I had outlined toward more or less at the beginning of my talk. It's not necessary to experimentally produce and crystallize these proteins to obtain information about their biology. We can focus on the important hypothesis and choose a relevant target to answer our biological questions. And with the support of AlphaFold, in a much quicker way, we can design targeted experiments to confirm their function, to challenge our hypotheses and formulate biological answers about the mechanisms of these targets. Of course, the tool has just been developed. It still has some limitations. It cannot work very well for large complexes like the ribosome or the polymerases, for which I showed the very first videos at the beginning of my talk. It's still not performing well for RNA or DNA structures. And it doesn't capture all the dynamics of these systems. Nonetheless, it's a first and very remarkable step to make it much easier for scientists to understand the function of biological macromolecules, and for non-scientists to get a grasp of what it means to be a protein, and how our bodies, our cells actually look like at very high resolution. I'm at the end of my talk. I hope that at minimum, I convince you that structural biology is a extremely fascinating area to explore. I've made one important point, that automation, online resources, in artificial intelligence tools make it now really high throughput, expanding the spectrum of processes that we can study, and expanding our chances of success to get really useful biological information. But importantly, I hope I also made the point that all these resources, which are available to the entire population, make structural biology, and therefore the essence of life, accessible to non-structural biologists, so that also students like you, who will be the next generation of decision makers, can appreciate that if we want to untangle the secrets of life, we have to make targeted investments and allocate resources to this sort of studies. I'd like to conclude by introducing to you what are instead my lab members here in Grenoble. We partly work on all the aspects that I have outlined in my talk. We determine structures of proteins, structures of large complex RNA molecules, and it's really a team effort if we can produce results in this sector using facing the challenges that the protocols imply, and making the best use of all this large infrastructure that is made available to us. I also want to point out that to be successful in structural biology, as I've already mentioned throughout my talk, one needs funding and the activity of even just a fairly limited small group like mine that counts around 5 to 10 people over the years requires contributions from many funding bodies. I hope I gave you a glimpse of what it means to be a scientist, and I'm happy now to interact with you if you have questions, curiosities, or if something was unclear in the presentation. Thank you very much for your attention.
Thank you, Marco. You heard a lot of claps here from the students.
That's encouraging, yes.
Thank you very much. Anyway, it's not easy to talk about a big part of biology in few minutes for us that we are not biologists and we know just few things about it. In our lessons we are talking about digital transformation, so it's very far feed from your. And I want to try to understand maybe better. You at the beginning you said you were talking about maps, saying if you have a map of a city, a detailed map of a city, you can know many things of the city, how the city works, how can we change some characteristics of the city, how it works. If I understood now, with AlphaFold, with artificial intelligence, we have like Google Maps of all the proteins. Is it right?
Exactly, exactly. All those movies I've showed you, it's like opening Google Maps, and instead of seeing the streets of a city, you see the amino acids, which means the building blocks of, you know, the houses aligned one after the other that compose a protein. Okay, and so you can navigate through it the same way as you navigate through a Google map to reach your target, to reach your destination. Okay, and so that's exactly the fascination of and what structural biology enables us to do. Yeah,
And
and AlphaFold just makes it much faster, you know, because now actually whatever biological question one has, you know, one can directly access, okay, through these online resources, the predictions of the structure, and of course with a little bit of uh, instructed experience, okay, with a little bit of knowledge of biochemistry and of the field, one can formulate accurate hypotheses. You know, if there is a disease and the disease, the doctors discover that the disease derives from a genetic mutation. Okay, sometimes you hear, you know, cancer because of a genetic mutation in a specific gene. Well, we can immediately look, what does this gene do? How is it converted? Into which protein is it converted? You remember my first slide, the central dogma, okay? A gene is converted to a protein. So if the gene is mutated, how is the protein mutated? And can we understand, where is the mutation? Can we understand what could be the functional consequences of that mutation from the structure? Okay.
and how much changed times in your work? A lot, but how
This
much?
changes time. So, you know, when we proceed experimentally, this is a very trial and error process. So the time can vary from several weeks to several years or decades for the very challenging proteins that we study. Okay. Now, AlphaFold makes this resource the endpoint of a weeks to months to year-long project immediately available, essentially. Okay, so we can start already from what was the end of a typical project until now. Okay, and move forward. Okay, so we skip, potentially skip months or years of experimental studies.
I think this is a perfect and beautiful example of how new digital technologies are changing every aspect of our life, not only in manufacturing or services on app, on websites. I don't know. They are changing everything. So also the scientific research and
Also the
development
scientific
of
research and also all our efforts to disentangle, you know, what I mentioned in my talk and I described in my talk as the secrets of life. Yeah,
yeah.
absolutely.
And do you think it was possible to predict 20 years ago these big changes?
No, in no way. 20 years ago, I told you there were already efforts to go towards this direction, but predicting that we would reach this stage so efficiently and to the scale that AlphaFold has enabled, I don't think that anybody in the field could have predicted this. And I see this from my senior colleagues here at EMBL, who are among the pioneers of structural biology as a field, as a branch of science. They are absolutely amazed and fascinated by these advances. Yeah.
So the reality is more than dreams.
Absolutely, absolutely. In this respect, it has really and actually it's reality, it's more than dreams, and it's fostering even bigger dreams, because of course now having seen the potential of these computational resources, of course we are already pushing this forward to break even more barriers. Okay, I mentioned about some limitations that are still out there in the system, and we certainly trying to break those limitations, solve those limitations, and make the tools even more powerful.
Anyway, some dreams for the future 20 years, knowing that now we have this kind of instruments, try to let dream the future.
But absolutely, you know, one very active line in of research in my lab deals with not with proteins, but with the intermediate stage of that central dogma that I described to you, the RNA. Because in the last few years, another revolution happened in science, that is that we sequenced the human genome, okay, just 20 years ago. And by sequencing the human genome, we discovered that only a very small portion actually is dedicated to the production of proteins. The majority of the genome produces RNAs that are also important for maintaining our cells and our body alive. And about this RNA, we still know very, very little, and we have big challenges in making photographs of how this RNA look like. So one of the big dream for us, for my lab in particular, is to convert now these artificial intelligence tools that are so successful for proteins and make them useful also to better understand how RNA works. And then we will have solved not just 1/3 of the biological world, but another important chunk, okay, another 30% of the complexity of biological macromolecules. And so that will give us again, empower us to treat diseases in ways that we cannot predict for now.
Okay, Marco. So thank you very much. Can you do something for the city of Rome with
Exactly. I'm not an expert in artificial intelligence design, but maybe you guys that are digital expert should take inspiration from this talk and from the biology that I shared with you today to, who knows, what invent something important in your field of research. It's a common line, right? Humanity, we have always tried to get inspiration from nature to improve our quality of life, you know, design planes on the image of flying birds. And so it's important to study the microcosmos, okay, the atomic resolution world, biological molecules, to get maybe inspired to develop nanomaterials, robots that work efficiently and precisely to produce new synthetic materials, to produce new drugs, to produce new tools and new mechanics that may facilitate life on Earth in some way.
Okay. So, Marco, thank you very much. Thank you all, the person, the visitor here in this lesson, our students here. This was the last lesson of this group of this edition of LUMSA Innovation Talks. Maybe we will have another one next year. So, Marco, thank you
very much for having me, and I'd be very interested to have your feedback if you can relay with Leonardo, and let me know any comments you have on how you perceived this experiment that is communicating biology to all of you. Good luck for your careers, and thank you for the invitation again. Bye-bye.
Thank you. Bye.